Perfect. Okay, well, thank you very much for joining us and choosing this workshop rather than the workshop to be a successful pharmaceutical physician.
We're already successful.
Yeah, it's almost a bit like the healthy volunteer bias, isn't it, which we'll be covering later, or survival bias, depending on how you look at your field. So my name's Debabrata Roy. I'm the head of epidemiology and research at the DSRU, and it's my colleague Ms. Samantha Lane, senior research fellow at the DSRU. And so in this workshop, we're covering methods, strengths and limitations, essentially a whistle-stop tour of some of the challenges that we face in pharmacoepidemiology, as well as obviously, its strengths and its limitations, and I suppose a lot of that context was set in the talk that we've just heard from Saad. So yeah, with no further ado, I'll hand over to my colleague Samantha Lane.
Thanks, Debs. So thanks for the introduction. I'll just give you a brief run through of what we will actually be covering in this session. So essentially it's going to be a very brief introduction to what is pharmacoepidemiology. So we will discuss some of the major study designs that we use in pharmacoepidemiology, including a description of each method and their strengths and limitations. We will look at some of the sources of data that we use and consider how we might go about choosing the most appropriate data source that we can. And we'll then move on to think about some of the limitations of pharmacoepidemiology in more detail, specifically the issue of bias and confounding, and how we might try to minimise their impact on our research. And then finally, we'll briefly discuss some of the overarching aims of pharmacoepidemiology studies. So what is pharmacoepidemiology? Pharmacoepidemiology is the study of the use of and the effects of drugs in large numbers of people, and it really combines the principles of epidemiology with clinical pharmacology. So we apply epidemiological techniques to study the use of drugs in large populations. And these studies are not interventional, rather they examine the use of drugs in their real-world clinical use. Pharmacoepidemiology can also be defined as the study of the frequency, distribution, and determinants of disease in humans, and in this case, the determinants are the drugs, vaccines, or medical devices. And the main goal of pharmacoepidemiology is to quantify drug usage patterns and adverse effects. Pharmacoepidemiology studies can either be descriptive or analytical. So descriptive studies characterise the distribution of an outcome within a population. So they're used to describe the distribution of an exposure or an outcome, to describe patterns of disease occurrence, patterns of exposure, or they may describe a relationship between a medicine and an outcome, but it doesn't really take it any further than that. Whereas analytical studies, they're used more to test hypotheses or to test the effectiveness or safety of a medicine. And they're used to determine whether statistical differences are present between a suspected causal factor and an event within different groups of people. So some of the main study designs that we use in pharmacoepidemiology are listed here on the slide, and we will go through these one by one. So the first design is case reports and case series. Now, you may have seen these in the literature. These designs are used to describe an event following a suspected exposure in either a single individual or a very small group of patients. And case reports and case series are very useful to look at trends, and they do provide a lot of clinical detail, but there is really no other context. So some strengths of case reports and case series, they're really useful to detect novel associations and generating hypotheses, so they're very good for pharmacovigilance purposes. And they have a high applicability when other research designs are not possible to carry out. They also allow an emphasis on the narrative aspect, which gives an in-depth understanding of what they're describing, and they have educational value. However, there is a lack of generalizability. It's not possible to make comparisons using case reports and case series, and there's no possibility to establish cause or effect relationships. So there is a danger of over-interpretation. They're also subject to publication bias, and they focus on the unusual. So the next type of study is the cross-sectional design, and this measures the frequency of an exposure or an outcome in a defined population at a specific point in time. Cross-sectional studies can either be descriptive or analytic. So descriptive cross-sectional studies describe the frequency of exposures and outcomes at the given point in time, while in the analytical studies, data is collected simultaneously for the exposure of interest and the outcome of interest, and the prevalence of the outcome is then compared for the people who were exposed with the people who were not exposed. Cross-sectional studies are particularly useful for defining the health needs of a population at a particular point in time. However, because these studies are based on the existing or prevalent cases of the outcome, these studies are less useful for investigating relationships than some of the longitudinal study designs that we will come on to discuss. Interpretation of cross-sectional studies can also be difficult as the direction of causality is not always clear. And by this we mean whether the exposure happened before or after the event of interest. So cross-sectional designs are useful because they are population-based surveys, so they gather a lot of data on a lot of individuals, and they're relatively quick and inexpensive to do. They can establish outcome or exposure prevalence estimates, which can be useful, particularly for public health and health planning. And they're also great for hypothesis generation. But they have a limited value for establishing relationships, they are prone to misclassification bias, and it's not possible to determine causality due to the difficulties in establishing the time sequence. And also it is important to remember that prevalence depends on the incidence of an outcome as well as the length of survival following the outcome. So moving on to perhaps one of the most common designs used in pharmacoepidemiology, which is cohort studies. So in a cohort study, you start by selecting a study population or a cohort of people who do not have the outcome of interest. You then establish each person's exposure status, and you observe the cohort over time to compare the incidence of the outcome in the exposed group with the outcome in the unexposed group. So this means that a cohort study is longitudinal in nature. Cohort studies can either be prospective, so looking forward in time, or retrospective, where we would use a historical time period. And to determine whether there is an increased or decreased risk of an outcome in the exposed compared with the unexposed group, we can use risk and rate ratios and odds ratios. So this diagram demonstrates a prospective cohort design. So let me see if I can get my pen. So you start here with your cohort of people at the start of the study, and you then group them into who has been exposed and who is unexposed. You then follow each exposure group over time to determine who developed the outcome and who did not, and you then compare the frequency of the outcome between the exposure groups. And a retrospective cohort study is done in exactly the same way, but it uses a historical time period. And it's important to note that the direction of inquiry is always forwards in time, even in these retrospective cohort studies. So cohort studies are very useful for rare exposures and for measuring multiple outcomes, and you can use them to calculate incidence risks and rates. They're longitudinal in nature, which enables long-term observation, and it also means that you can develop these temporal relationships between the exposure and the outcome. And the methods for measuring exposure and outcome in cohort studies are less prone to bias than in some other designs. However, they are insufficient for rare outcomes because they would require a really big sample size. They're also liable to bias and confounding, and limiting the effect of bias and confounding requires effective study design, conduct, and data analysis. So next up is the case control design. So in a case control study, you start with a group of people who has experienced the outcome of interest and a group of people who have not developed the outcome. So those with the outcome become your cases, and those without the outcome are your controls. So starting off with a group who have the outcome means that the case control design is particularly useful to study rare events. Once you have established your cases and controls, you then look back in time to determine who had been exposed to the exposure of interest and who had not been exposed. And you can then compare the frequency of the exposure in the cases with the frequency of the exposure in the controls using an odds ratio. So this diagram demonstrates a case control study. So here's your study start. You start with your population, which is made up of cases and controls. You then look back in time to determine who had been exposed and who had not been exposed, and you can then compare the frequency of the exposure between the cases and controls. So case control studies are relatively quick and inexpensive in comparison with perhaps cohort studies, and they are useful for rare outcomes. They require a smaller sample size than cohort studies, and they're suitable for outcomes with a long latency because you're looking back in time. However, they are particularly prone to bias, especially recall bias, and they can't be used to calculate incidence risk or rate as the denominator is not defined. And it can sometimes be difficult to establish a temporal relationship between the exposure and the outcome, which is important to bear in mind. Similar in design to the case control is the nested case control study. And this is essentially a case control study which is nested within an ongoing prospective cohort study. Again, these studies are useful for rare exposures or events. And in a nested case control study, a subset of controls from your cohort are compared to the incident cases of the event that you're interested in. Controls are selected from relevant risk sets and are usually matched to cases on one or more characteristics, most commonly age and sex. And nested case control studies are an efficient design to answer questions. They have a reduced cost and time because exposure data is only collected for the cases and the controls. So moving on then, we have case-only designs. And these include people who have experienced the event of interest at some point in time, and individuals act as their own controls. So this means that comparisons are made within individuals, so the exposure period is compared with an unexposed period. And confounders which are not affected by time are therefore removed by design. Case-only studies are usually conducted using retrospective data from large databases, for instance, using electronic health records or health insurance claims databases. One type of case-only design is the self-controlled case series. This is a good design for acute events, transient exposures such as vaccines, exposures with widespread usage, and for when suitable controls cannot be identified. There are some assumptions which apply to self-controlled case series studies, including that occurrence of an event should not affect subsequent exposure, that event rates are constant within intervals, and that events must be independently recurrent or rare. If some or all of these assumptions cannot be met, it is possible to apply sophisticated analysis and modelling methods to control for this. So the study designs that we have looked up until now are all individual level studies, meaning that exposure status and the outcome were compared at an individual level between the groups. However, we have this design called ecological study design, and these are different because we compare exposure and outcomes at a group level. We might want to use an ecological study to investigate differences between groups, maybe to investigate group level effects, or simply because groups are more convenient to study. However, you need to be careful about a type of bias called ecological fallacy, and this is where results are misinterpreted and inferences are made at the individual level. And finally, we have 2 study methodologies which have been developed by the DSRU to specifically study the use and safety of medicines in the post-authorization setting. These designs are called Modified Prescription Event Monitoring, which investigates medicines used in primary care, and Specialist Cohort Event Monitoring, which examines medicines used in hospital or specialist settings. Both methods involve the research team contacting healthcare professionals who are involved in individual patients' care to obtain details on the effects of the treatment under study. These studies are designed to monitor the use and general safety of medicines and can identify potential new unexpected risks of specific medicines under real-world use in clinical practice. So I have a little exercise now where we'll run through 3 study designs and see if you can come up with which design it is. So this first one, we start with a group of people, which is your study population. You then classify them according to the presence or absence of the outcome, and then you compare the frequency of the exposure in the people with the outcome compared with the people without the outcome. So, I don't know, just shout out. Yes. Brilliant, yeah. So next, you start with a group of people who do not have the outcome of interest. You classify them according to their exposure status and you observe them over time. And then you compare the frequency of the outcome in the exposed with the frequency of the outcome in the unexposed. Yeah. And then finally, you start with a group of people and you compare the prevalence of the outcome in the exposed and the unexposed at a given point in time. Yes. Okay, great. So we'll now move on to consider some of the data sources which can be utilised for pharmacoepidemiological research. So there are many data sources which can be used for conducting pharmacoepidemiology studies. And the decision on which data resource to use depends on a number of considerations, including the study objective, the possibility to capture the exposure and the outcome of interest, the sample size, and the time window of the data source. And it's very important to know the strengths and limitations of the data which is being used. So data used in pharmacoepidemiology can be split into broad categories, either primary or secondary data. So primary data is data which is generated specifically for the research study, which is useful because you have control over what data is collected. However, primary data collection is time-consuming, it's expensive, and it limits the number of people that can be included in your study. Secondary data, which is data that has been collected for a purpose other than research, is commonly used. So some common examples of secondary data that we might use in pharmacoepidemiology are patient medical records, which can be obtained either using survey of healthcare professionals or using databases of anonymised electronic healthcare records. Health insurance claims forms and billing can also be used. And registries are another potential source of secondary data. So registries either focus on a disease or a drug, and they usually involve recruitment of all patients who either have that disease or are exposed to the medicine of interest. And a registry involves the systematic and ongoing collection of data with defined outcomes, exposures, patient populations, and period of time. So this can be a really useful source. So some strengths and limitations of secondary data are here on the slide. So secondary data provides real-world data at an individual patient level. It's usually prospectively collected and time-stamped and gives an unbiased large sample size. However, there are some limitations. So secondary data is routinely collected data and it's not necessarily for research purposes. And that kind of introduced a few limitations of its own, which can include completeness of data, perhaps it doesn't contain all the variables that you need, the population coverage can vary, and the quality of data can vary as well. So when choosing a data source, consideration should be given to whether the data source contains the data on your population of interest. So which patients does it contain? Are they representative of the population of interest? And is there a big enough sample? And you also need to consider whether you can gain accurate information on the exposure of interest. For example, if the drug is only prescribed in secondary care, then you would need to use a secondary care data resource. Additionally, consideration should be given to the level of data on the exposure, such as the timing of use, construction of treatment episodes, and assessment of cumulative exposure. You then need to think about whether the data source contains enough information about the outcome of interest. So you will need to know what happened and when, and consider things like how the outcome is defined, how the data is captured, and if there is structured coding, and any data validation. The most appropriate data source will also contain data on all potential risk factors. So Debs will talk a little bit about confounding factors later on, but for example, these may include things like lifestyle factors, comorbidities, and concomitant medications. And finally, some important considerations are database issues, including data quality, coverage of the data source, including the population and time period, any accessibility issues, including permissions and cost of accessing the data, and how up-to-date the data is. And finally, it's important to think about the health system and how this is organised and reimbursed, as that may influence your choice of data source. Sometimes we may decide that multiple data sources are needed to perhaps increase the number of patients or the representativeness of the data and the breadth of patient information. However, you always need to be careful when using multiple sources of data, as there may be an overlap between them. And this could result, for example, in duplication of patients, as can be seen when using healthcare records from the data sources CPRD Gold and Thin. So in addition to those that we've already spoken about, other data sources used in pharmacoepidemiology include spontaneous reporting databases, for example, the UK's Yellow Card Scheme, social media, and big data. So spontaneous reporting databases contain adverse drug reaction reports from patients and healthcare professionals. These are surveillance systems employed throughout the medicine's life cycle, and are generally used to monitor the safety of medicines and for the detection of signals of emerging safety concerns. However, there are important limitations for this data source, including underreporting, selective reporting, variable data quality, and coding practices. And importantly, there is no denominator data, so it is not possible to estimate the incidence using spontaneously reported data. Okay, so are there any questions at this stage? Yep.
You mentioned social media there, and Saad mentioned it earlier as well. Is there, and we know there is gigabytes of data out there. Has anybody had any success in looking at social media or any projects looking at social media as far as this is concerned?
Well, yes. There is now maybe 10 years now where the MHRA, in collaboration with various European entities, regulators, have the so-called the Radar project, where they specifically had teams to look at social media and the advantages of social media. And as I said in my presentation, if you Google social media and pharmacology, and people find that patients report adverse reactions in social media different from those who reply to doctors. For example, thinning of the hair, gaining weight, impairment of sexual libido, and things like that. So there are groups of events which people in social media talk about compared to what they report to their doctors. And I always challenge people to say, is there an important signal which led to withdrawal of a product or major restriction of use of a population of use came from social media? And that has not come yet. The other thing which people are really spending a lot of money in using artificial intelligence and machine learning to try to identify patterns from the narratives of social media. We know, for example, in radiology, you use artificial intelligence to relate the image with the lab results, with the histology, with everything, and come like with different stages of tumor staging. Social media, because of the paucity of information and the poor quality of information, is yet to come to that. But if you're asking me how many startup companies have been funded by venture capitalists to try to identify patterns in social media, you'll find a lot. So don't let my skepticism color your ideas. It's work in progress.
And I'm just aware that there's got to be a lot of information out there. It's how useful is it, and at some point will the MHRA.
And just one last thing to say: social media is two types. The type of general social media between friends and so on. But then, I mean, I discovered after my event 3 weeks ago that there is a baker's Facebook group, which gave me a lot of information about the duration of my illness. Some of it, like say, use smear your leg with castor oil and things like that. And some of it is useful. So social media is a hodgepodge, but there is some useful information.
I just have a follow-up question to that, actually, out of interest, with regards to the vaccine side effects, or particularly VIT, because I know that obviously there was spontaneous reporting, of course there was, of these kind of incidents, but there was also an awful lot going on Twitter and the haematologist WhatsApp. So I just was interested in your take on how much of a contribution that actually made to safety slogans.
Well, I don't want to take over, but one of the hallmarks of the monitoring of the vaccines, all the methods which Sam has talked about, the main decisions about how to deal with anaphylaxis, how to deal with the thrombosis or thrombocytopenia, how to deal with pericarditis and myocarditis with the mRNA vaccine, all of them were based on spontaneous reporting. The different spontaneous reporting with COVID-19 vaccine, there were a lot. The MHRA gets 45,000 reports a year. During the COVID years, they get 450,000. They engaged an artificial intelligence company to look into that. And all the decisions, some countries, like Denmark, decided not to have the AstraZeneca vaccine based on a small number of reports, a spontaneous report. Spain restricted usage in various age groups. So there was disproportionate usage of spontaneous reporting. Excuses, there was no other. We in pharmacoepidemiology produced results in June and August, while the decisions were made in March and April. Spontaneous reporting with all its limitations. But the good thing about it, we say in spontaneous reporting we have inaccurate numerator and an inaccurate denominator. The denominator was accurate. You know daily how many people had the vaccines, and the numerator had more information. So it just, we are now going to spend the rest of our lives in the futile activity of trying to learn from that to be prepared for the next. But I don't think. But if you're asking me, the contribution of social media, Facebook and Twitter, to the regulatory decision making, I didn't see any of them mentioned in the official communications. Did you see that? No.
No.
Yeah, I did.
Yeah.
At least I know one case when the recently nominated Ministry of France, of the Health, Ministry of Health of France, tweeted that ibuprofen was had been published in Lancet, like causing a worsening of COVID cases. All the agencies reacted and advised against the use of ibuprofen worldwide.
Ibuprofen. Yeah, we're talking about. Okay.
So,
Yeah.
and this was a tweet.
No, we're all now in the learning phase of what has happened.
And it was I suppose quite a unique sort of situation we were facing at the time. suppose, quite grateful for any sources of information, really, weren't we?
But back to my point, I was frivolously saying, there are social media specific for diseases, inflammatory bowel disease, this leukemia, that lymphoma, whatever. And that is different from the sort of friends talking about what did they have, for dinner last night.
Yep.
Oh yeah. So is it MPEM and SCEM?
Mhm.
What were the differences between the two again? And so they were created by the DSIU, is that right?
Well, event monitoring is a wider.
Yeah. So I guess PEM came about. It started off as prescription event monitoring, sort of before my time at the unit, which then sort of evolved into modified prescription event monitoring. And I suppose the sort of main difference is, I suppose, who we're targeting when we're sort of sending out the questionnaires. They're both essentially non-interventional methods, which are one of its strengths. With modified prescription event monitoring, we are able to identify the GPs who are prescribing the product, and then obviously we can target them with questionnaires, bespoke questionnaires, and electronic capture forms. And with the specialist cohort event monitoring, as Sam mentioned, it's more the specialist, the secondary care, private sort of healthcare practices.
So one of them, PEM is drugs used in the communities by GP, SCEM is drugs used in hospital,
Yes.
not necessarily inpatients and outpatients, but specialists.
Yeah, and even SCEM studies can have a level of involvement from the GPs as well. Thereafter, if they're released back into the community and follow up with the GP, we can obviously do that as well. So it can be a sort of combination of both if it's required.
I'm going to ask, I think, what a provocative question is, how good do you think we are, or you are, at signal detection? And do you ever challenge yourself and simulate data to see if you would have picked it up, or is there any kind of metric that sort of says, this is kind of the level of threshold or the number of people that would be affected in order for us to detect a signal?
There's a guy called Andrew Bate, who now works for GSK, who used to work for the Uppsala Monitoring Center. He's one of the gurus of signal detection. And he contacted me about 3 months ago, asking exactly the question which you asked. He said, the time has come for us to try to do an international audit to see whether our sophisticated systems, observed versus expected, disproportionality, or whatever, what they have detected, which couldn't have detected conventionally, that they detected earlier than could have been done by other, and that they detected in special population and found special characteristics for it. And we have an international working group on advances on pharmacovigilance, and I think your question needs to be answered. Because otherwise, if we can't prove it, so that question has not been formally in a big brutal picture, which you're saying and Andrew's saying. I think the time has come for us to answer it. And plus also, the role of AI and machine learning, and that what value addition they're adding. So, but this is like 3 PhDs.
Yes. You're looking for someone.
Sorry, you're saying something.
What approach would you think to use to make that analysis?
Well, first we
Retrospectively,
were going to do actually, as Sam might do that, for a Delphi exercise. We asked all the signal detection gurus these questions, see what they say, and then we could follow it by a systematic review in the literature, and then we could do inquiries from the regulatory authority. So as I say, to answer this question, you need at least 3 years of work. Whom do you work for? Whom do you work for?
Oh, very small company. They've never paid.
So they don't give grants for methodological research.
You send me a request and run it.
No, but we need to get a grant to do that study, and I think we
But
need to.
it's really interesting, isn't it?
Probably get it from the Wellcome or the Nuffield or place like that.
Yeah.
But also, and it goes back to what you said about biologicals and small molecules.
Yeah.
The difficulty of signal detection is
Yeah.
different. So
Oh, yeah.
even when you assess a set of clinical data, you have to keep in mind the clinical need, the size of the population, and what is your chance of detecting a signal.
That's why all of you, to be successful physicians, you can become successful pharmacovigilance physicians. It's easier. The competitor there is stronger, the competition.
So conscious of time, because we've got another half an hour left, and obviously we can hopefully have some time for questions at the end. But it's a continuation of the workshop. So Sam's obviously presented all the different study designs and different methodologies that we use in epidemiology and pharmacoepidemiology, as well as the different data sources. I'll be touching on, I suppose, what are the inherent issues with epidemiological studies, how they can be minimized or should be minimized if you're obviously reading a manuscript that somebody else has published. So you can obviously critically appraise studies rather than just accepting conclusions. And obviously how to handle other aspects which are just natural to the field of real-world evidence, such as confounding, and how that's kind of handled in the analytical stages. So we've touched on, or Saad touched earlier in the talk about validity of studies. Internal validity is essentially, is what gives us confidence to make conclusions about the study that we've conducted based on the data that we've used and on this, small sample that you've managed to kind of use. Whereas external validity is then provides us with that confidence to be able to make those inferences from your little study that you've done about the source population, because that's what we're interested in, rather than just your N of 20,000. So that's your external validity. I'm sure you've heard of generalizability and other terms like that. So essentially, what we're trying to do in epidemiology is to get to that truth. We're very conscious about that our findings are as close to the truth as possible. So in that sense, a valid inference is one which or a perfect inference, or maybe valid is not the right word to use, but a perfect inference is one which ideally has no bias. So in observational studies across the years, you may find random associations, and these may have essentially come about because of bias in the study design, in the study conduct, maybe because of confounding, maybe it wasn't handled correctly in the data analysis. So these are things that you need to be aware of in real-world data in observational studies. And unfortunately, if you're based in the UK, you know this is a very quality newspaper in this country. But unfortunately, the media is known to blow studies out of proportion and out of context. They may be reporting findings from studies which have bias and confounding, and studies have shown that both positive and negative news stories about medication can affect usage in the population. So we talk about data error, and we need to, I suppose, ascertain whether it's a natural variation or whether it's actually a genuine mistake. So in statistics, an error is defined as a difference between a calculated or observed value and its true value, and that's the true value which you would assume from the population if you had it. Random errors are essentially results which have a sort of a natural fluctuation in the observation. It's kind of a natural random variation. We're on the slide, yep. And systematic errors. These are introduced through inaccuracy in the system, and they occur reproducibly. And what I mean by that is it's something inherent in the study design, something inherent in the study conduct, which is causing your results to lack accuracy. And you can see the picture there. So so if you imagine a study giving you a set of results, if you introduce some random variation to that, oh, so I'm going the wrong way, there you go. So what it does, it affects your precision. You end up with imprecise results. And ways of minimizing that is to increase your sample size, increase the power of the study, and you can get more and more precise point estimates, or whatever sort of parameter you're measuring. However, with systematic error, otherwise known as bias, is that coming through? Yeah. Okay. So you end up with inaccurate results. So every time your results are inaccurate, and you can see. Two studies. Even if you had a large enough sample size, because of the systematic error in that study, you end up with inaccurate results. So the definition of bias is any systematic error in the design and or conduct of the study. In observational studies, it can result in potentially an incorrect association measure between the exposure and the outcome, and it's challenging and in many cases not possible, and it does depend on the type of bias, to deal with this at the analysis stage. So I think in epidemiology, I suppose in any branch of medical research, it's very important to correctly design as well as conduct your study to minimize your bias. And often this may not be feasible based on the type of study you're doing, in which setting it's being performed, and it may be for hypothesis generation, for example. We may not have the funding to do it in a different way, but it's very important to be aware of the different types of bias, so then you can obviously interpret your results in the proper context it should be interpreted in, which I think is always a problem. Okay, so I'm just going to cover a few different types of bias, just provide like a brief description of each. There are other types of biases as well. I'm covering just, I suppose, the main types. And I guess, a lot of these biases may be dependent on the study design that Sam covered in her talk, and it may not be relevant to a different study design. So if we cover first, so we've got selection bias there and all the different types, information bias, covering two types there, and it's information bias which then leads to a type of bias known as misclassification bias, which then can be differential or can be non-differential, and that's something you may come across if you're reading, for example, a limitations section of a publication, and they'll talk about whether the bias, there may be bias in the system, but it is non-differential, which sort of reduces its harm somewhat. Okay, so selection bias is especially an issue for those case control studies which you were able to identify on the questions, where cases and controls are selected. So this bias arises when the chance of getting into the study sample from the source population, and whether that's related to whether it's related to both the exposure and the case control status. So there is a different chance of being recruited depending on that relationship between the exposure and whether you're a case, and the relationship between the exposure and whether you're a control. So a form of this is called Bergson's bias. So it's an issue with hospital-based case control studies. The cause is that hospital cases and hospital controls are different from one another because the combination of being exposed and being a case increases the likelihood of you being admitted to hospital. So what you then end up seeing is a systematically higher exposure rate amongst the hospital cases. So you can see here is a bias in the selection of the cases here. So you've got a household study and you've got the association between respiratory disease and osteoarthritis. And you can see, obviously, it's like a standard 2x2 table. You can see odds ratios here being calculated because it's a case control study. And underneath it, we have a hospital-based study looking at the same association between respiratory disease and osteoarthritis. And you can see here the differences in the proportion of people in the two studies. The household survey shows an odds ratio of 1.1. It's sort of suggestive of a weak association, whereas a hospital survey shows an odds ratio of 4.1, suggested of a strong strong association. So this difference is due to the chance of recruitment for exposed cases is much higher in a hospital study than the household study. So essentially what you need to do to minimise this is to make sure that the source population from where you're recruiting your patients is the same for both cases and controls. Okay, another bias is in the selection of controls, and it's this was shown in the Dole and Hill study where they looked at the association between smoking and lung cancer. The control group were chosen who were patients in hospital without lung cancer, obviously, that's their controls. However, the control group was shown to smoke more heavily than the general population. So the control group should have come from the same source population where the lung cancer cases arose. However, as I mentioned, there are challenges in selecting the control group, and that can be due to restrictions on where your study is actually being conducted, could be due to funding issues, and it's just something that you need to take into note when you're interpreting your findings. Okay, another form of selection bias can be your healthy worker volunteer effect. This can affect all study designs. Different characteristics, so healthy workers and volunteers have different characteristics to the general population. Healthier people we know tend to volunteer for studies. Healthier people tend to be and remain employed, and they'll have a different set of risk factors compared to unhealthy people, or maybe even normal people. And this can affect your external validity as well as the generalizability of your results. Another form of selection bias is survival bias. This can affect all study designs again, and it results from an improper sampling of cases, and it occurs if the risk factor if the risk factor, the exposure of interest, whether that's associated with death from the outcome being studied. So for example, if you're looking at the association between smoking and myocardial infarction, heavy smokers who have died from MI, they will not be recruited into a prospective cohort study as they're no longer alive. So then the remaining MI cases that you do see will show a lower frequency of smoking as the heavy smokers are no longer around. This may not be, and it shouldn't be, an issue if you're doing a retrospective cohort study. They're all in the database, so you've just got to be, and I think this can be, again, handled in the study design phase. Make sure you've got the correct data source, you're using the correct approach to study this association. Another form of selection bias is detection bias. This is when a medically related exposure leads to detection of the study outcome. And this can be particularly when it's a disease that has a high proportion of subclinical cases. So for example, if you're looking at the association between erectile dysfunction drugs and skin cancer, so you may have a hypothesis, is there an increased risk of skin cancer in men prescribed erectile dysfunction drugs? However, there may be a selection detection bias here where these men are subject to an increase in medical surveillance because obviously they need to go in and for their repeat prescriptions, and due to that increase in medical surveillance for this group, you're able to detect cases of skin cancer, and you have to be aware of these forms of detection bias. So minimizing selection bias, sample the controls from the same source population where the cases arise. Then essentially you've got the similar sort of distribution of factors in both cases and controls. Control should represent the source population in terms of exposure. And you can also work to keep non-response to a minimum. And you can also make efforts to characterize non-responders or characterize those who may have a loss of follow-up, who have dropped out of the study, and you can help characterizing those patients. You can get an idea of whether there's sort of factors behind the non-response in the study. Okay, so I've covered these different types of biases, and I mentioned that information bias leads to misclassification bias. So information bias, so this results from, or it can result from, imperfect definition of the study variables, whether it's a misclassification of the exposure status or whether it's a misclassification of the outcome status. And this can affect different study designs. In retrospective cohort studies, whether the correct ICD-10 codes are being used, whether the correct Medcode IDs are being used in CPRD, for example, to ascertain exposure or outcome status, and it can also arise from if you have flawed data collection procedures. So it may result in misclassification of exposure and or outcome status. As I mentioned before, information bias causes misclassification bias, which can be either non-differential or differential. So recall bias can arise, especially in case control studies. If you're not collecting information, if you're collecting information directly from participants, then a subject's recall of their prior history or their prior exposures may vary as to whether they're a case or a control. If they're a case, they may have a much better recollection of their prior exposure and prior medical history. And it's an example of a case control study looking at congenital malformations where during pregnancy, where participants may recall drug use differently. Another problem could be with interviewer bias, and this could be introduced directly by the study investigator who may be collecting the data for the study, and where information on exposure status then goes on to influence the classification of the outcome status. So interviewers may record information differently according to whether they already know a subject is a case or a control. These forms of bias can easily be minimized through proper training or even blinding. So as I mentioned, misclassification bias leads to two different forms. One can be non-differential. And this is when the error in determining the exposure status, but this error occurs equally for both cases and controls. So it could also be an error in the outcome diagnosis, could lead to false positives or false negatives. But again, the error occurs equally for the exposed and the unexposed group. And in this case, the effect on the measure of association is that it brings it down towards the null value. With the differential form of this bias, it's dependent on whether you're a case or whether you're a control. And be it that error, if it's in the exposure classification or whether it's an error in the outcome diagnosis, then it will depend on the exposure status, or maybe it's apparent in the exposed group and not in your comparator or your unexposed group. So there's a differential there. And the effect on the measure of association is that the bias can go off in either direction, so that your actual estimate could be higher or lower than it actually should be. So dealing with misclassification bias, I've mentioned some of these already. You can obviously minimize recall and interviewer bias. You can use accurate diagnostic techniques for the outcome assessment, and you can go on to screen controls for subclinical disease. So we've got about 10 minutes left, so I'm going to quickly go through confounding and also the strengths and limitations. So confounding is essentially, it's a natural part of, or an obvious part of real-world data. There are many risk factors that can be associated with an outcome of interest, and you can see with the word to confound, to cause, to become confused, to mix up. But it's essentially the effect of that extraneous factor that either completely or partially accounts for the apparent effect of the study exposure that you're interested in, or that masks an underlying true association. And it's something which is not directly connected with or related to something. And as I mentioned, it's a natural part of the study, so it belongs to the study. However, unlike bias, you can adjust for confounding in the analysis stage, providing you have the information on those confounding variables, obviously for all the participants in your study. And by adjusting for it, you're essentially removing its effect from the association you are interested in. So the confounder may account for all or part of an apparent association. It can cause an underestimation of that measure or an overestimation of that measure. A confounder may be another risk factor for the outcome. It could be a protective factor if you see an inverse association, and it could also be a surrogate for another risk factor for the outcome. And that sort of leads you on to how do you go about identifying a potential confounder. And traditionally, in sort of introductory epidemiology courses, they cover these kind of basic 3 rules. So the potential confounder, so this kind of triangle shows that relationship between the exposure of interest and the outcome, and that confounding variable sitting there. And you can see the arrows between the exposure and the confounding variable are two-way, whereas the direction of the arrow between the confounding variable and the outcome is in one direction. So here we have the potential confounder must be associated with the exposure of interest in the control group, so those without the outcome. The potential confounder must be independently associated with the outcome of interest, and it must not be on the causal pathway between the exposure and the outcome. So how do you go about identifying the potential confounder? You need knowledge of the subject matter, you need that clinical understanding, and you'll be able to identify confounders with that knowledge. And then the second step thereafter is with data analysis. So you examine if the potential confounder satisfies those 3 rules. And then at data analysis, essentially what you're doing is you're stratifying the data by that potential confounder, be it age or gender or a different risk factor, and that essentially removes the effect of the confounder. And you could examine what happens to the magnitude of the association in each of those groups of the confounder, and you can essentially answer, is the measure of association in each strata similar in magnitude and considerably different from the crude association. So we've mentioned prevention in the design stage of a study. So with respect to confounding, it can be prevented in the design stage of a study. And this can be done by using, obviously in RCTs we use randomization, Whereas in observational studies, we can restrict, so you can restrict to a particular subgroup of interest. However, restriction will reduce your sample size. And we can also use matching techniques, and that's where we match, and you may have heard of this, where a study you've matched on age, you've matched on gender, you can match on influenza vaccine history, you can match on a whole host of different factors. But you have to be careful with matching, is that you're essentially taking out that confounding variable, so you can no longer analyze that confounding variable and see the association that that variable has with the outcome of interest. So you have to be careful when you match as well. Or you can control the analysis stage, and obviously there's a lot of many different statistical software packages available, such as Stata or R or SAS that people use. And you can obviously use regression models, multivariable regression models, or logistic regression models, or use propensity score techniques, which are commonly now used in pharmacoepidemiology. Quickly, but propensity scores are basically where you take into account the factors which are leading towards a patient having that prescription in the first place. And by using this propensity score method, you take out those confounding variables which from the actual study, because you've already assessed it in the propensity for that patient to receive a certain form of treatment. But there's a whole host of information out there on that. So this just essentially shows you what's happening in a regression model. It's essentially an extension of the 2x2 table that I showed you. This is the mathematical formula for it, so I'm just a bit conscious of time. And you can see what's going on here. This is for a simple linear regression model, and you're putting each confounding variable into it and adjusting. So each factor on the right hand side of the equation is controlling for all the other variables on the right hand side of the equation. So you're adjusting for it, and you can then focus on the outcome exposure relationship. We've talked about this. You should beware of the black box, where, for example, if you include too many factors, it can result in something called over-adjustment. We've covered residual confounding. This is where, for example, there may be confounders that you haven't measured and you don't have information on, and that can result in residual confounding in your study. Okay, so touching on the strengths and limitations of pharmacoepidemiological studies, and I suppose we've covered many of these in the two talks already given today, but we've got large sample sizes, and with those large sample sizes we're able to quantify risks, which you're not able to do using spontaneous reports. With large sample sizes you're able to detect rare events. Real world use includes patients with comorbidities and co-medications and subgroups which are often excluded from clinical trials. You can look at products with wide scale or limited usage. You can look at short or long-term medication use. You can have a long follow-up of patients if you're looking at long-term effects. Many different databases, and you can have multi-database studies now where you pool results together. You can have studies all across the world in different European countries, for example, where you're pooling results together using a variety of global data sources, and these studies often use common data models to make sure that there's some harmonization of the data from all these different sources. And it can come from electronic healthcare records or healthcare claims data, or disease therapeutic registries. And we're constantly seeing improvements in IT hardware and software and statistical analysis methods, which is why we're seeing such a big sort of discussion into machine learning and artificial intelligence, because the technology is improving so much, because IT hardware capabilities are improving so much. We have more power, more RAM to actually process very, very complex scripts on very, very large sample sets. Limitations I've mentioned, obviously subject to bias. So when it comes to study design, or when it comes to critically appraising studies, be aware of the biases and how they've been handled in the studies. residual confounding, have all the confounders been handled appropriately, the generalizability of results. If your study's conducted in Japan, for example, are those results generalizable to the area of interest for you? Look at the quality of data, how it's been analysed, whether it's been correctly analysed, as these may impact the results and interpretation of findings. Have they conducted any sensitivity analyses to see how robust the parameters and the criteria they have used have been? And also, limitations. It does take time to conduct some of these studies. If you're looking at a retrospective cohort study or even a prospective cohort study, it's dependent on the data being available at the end of the day. Even, for example, with a case control study, you need to have that exposure outcome, to have been available for a while. We can have a reliance upon spontaneous, and this is, as Saad has mentioned, this has been shown to us in this initial stages of the COVID pandemic, where we have been reliant upon the spontaneous reports because we've been setting up those observational studies, and obviously, it takes time to accrue data, usage data for safety events to be reported, and all of that takes time. So we've touched on the aims of these of pharmacoepidemiology studies. It's traditionally used in post-authorization safety studies to monitor and evaluate the safety of medicines after they're improved. And these are always listed in the risk management plan. Can be used for drug utilization studies, which are very useful if looking for supporting new licensing applications, for example. For the continued performance and safety of products once they're on the market across the life cycle, evaluating the safety and effectiveness of existing medications for new indications, increasing role to support regulatory decision making to speed patient access to new treatments, and that's what Saad discussed earlier. And I suppose within the COVID-19 pandemic, we've discussed this, but as I mentioned, spontaneous reporting system and ICSRs provided that data on the emerging safety signals, but we're in a position now where we have ongoing and very flexible real-world evidence-based PE studies with evolving protocols, with multiple reporting timelines, and these will essentially help us to fully quantify and characterize these risks which have been associated with the COVID-19 vaccines and the COVID-19 treatments. And I think that's one positive sort of, development from the COVID-19 pandemic is the flexibility that we now have in these PE studies, that it could be based on a spontaneous report, it could be based on a signal being detected, that the protocols then evolve across the life cycle of these studies. So in summary, obviously, there are different types, causes and impacts of bias in pharmacoepidemiological studies. These can then go on to have an effect on the measure of association or the generalizability of results. So you should obviously plan to remove or minimize bias in the study design or conduct stages. Confounding is an important part of the study. It's a natural part of any observational study, and there's obviously ways of handling it, either through design or adjustment in the analysis stage. Pharmacoepidemiological real-world evidence studies utilize real-world data and are increasingly being used to support these regulatory decisions in existing and evolving safety and licensing scenarios. The strengths and limitations of PE studies, especially in the context of clinical trials. However, results from clinical trials, signals from spontaneous reporting sources, and results from pharmacoepidemiological studies are complementary, and they're complementary in the generation of a continually evolving evidence base across the life cycle of a medicinal product and its evolving benefit risk assessment. Okay, so we're 2 minutes over. Just whether there were any questions at all. Yeah.
How common these days are post-authorization efficacy studies?
How common are post-authorization efficacy studies? That's a good question. I think they are conducted and they are requested by the regulators. I wouldn't be able to tell you how common they are. I don't think they're not as common as post-authorization safety studies. Sar, do you have a take?
I agree with you. They are not as frequently done as people thought when they were first brought in, but the regulators are talking about increasing requirements for them.
Yeah, you can see we stick to the safety aspect at the DSI. Okay, well thank you very much.